Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. So, what we have seen in the previous lectures, in the last two lectures of biochemistry of lipids, so we have seen this lipids introduction, classification. Then after that, we have also seen properties of fatty acids, reactions of fatty acids, as well as importance importance of that essential fatty acids. Okay, we have after that seen different quality control parameters for the evaluation of quality of lipids and oils or fats and oils. That means iodine number we have seen, saponification number we have seen, iodine number, saponification number, acetyl number, Polinsky number, Richard Michel number. Yeah, those quality checks are very important. Again, we have also seen the phospholipids, their types of phospholipid, importance, functions of phospholipid, and other complex lipids. Okay. We have also seen the physical properties of lipids. Then we was discussing last time this cholesterol. So I would like to start from cholesterol only. So nowadays there is a lot of noise which are going about the cholesterol in the market. A lot of people, they are discussing the cholesterol, all that. So try to understand each and every single thing about the cholesterol. And again, very important for GPAT and upcoming exams. So GPAT में भी already ये questions पूछे हैं. So what is meant by cholesterol? So it is very important steroid or very important sterol in the human body. The molecular formula which was already in an exam. What is the molecular formula for cholesterol? C27H45OH. Remember the formula C27H45OH. And the ring present in the cholesterol. कोलेस्ट्रॉल के स्ट्रक्चर में जो रिंग होता है उसके ऊपर भी क्वेश्चंस आ चुके हैं. कौन सा रिंग होता है कोलेस्ट्रॉल में? रिमेंबर, साइक्लोपेंटेनो पर हाइड्रोफेनेंथ्रीन. The nucleus present in the cholesterol structure. Cyclopentano per hydrophenanthrin. Okay. Now there are some hydroxyl groups. There is one hydroxyl group at a C3, carbon number 3 के ऊपर एक hydroxyl group है. There is a double bond between carbon number 5 and 6. नंबर 5 6 के बीच में डबल बॉन्ड है. There are again two methyl groups at a C10 and C13, and there is a 8 carbon chain attached to a C17. These are the features. इसके ऊपर भी क्वेश्चन आ चुका है. कितने हाइड्रोक्सिल ग्रुप है? So there is a one hydroxyl group at a C3, one double bond between C5 and C6, two methyl group at a C10 and C13, 8 carbon chain attached to 17 carbon atom. So molecular formula, the heterocyclic ring present in the structure or nucleus, and features. Okay, now cholesterol, physically it is a solid. Cholesterol is known as a solid bile. Okay. Cholesterol occurs as a white or faint yellow, which is odorless, insoluble in the water, sparingly soluble in the coal, soluble in the organic solvent. जो organic solvent से में soluble है स्पैरेंगली सोल्यूबल होता है अल्कोहल में, इनसोल्यूबल होता है वाटर में, ऑर्डरलेस होता है, वाइट और फेंटली येलोइश होता है ना, सोल्यूबल इन द ऑर्गेनिक सॉल्वेंट्स एंड अदर वेजिटेबल ऑयल्स, मेल्टिंग पॉइंट इज अराउंड 147 to 150 डिग्री। ये कहाँ पे पाया जाता है, व्हाट इज द सोर्स एंड ऑकरेंस ऑफ दिस कोलेस्ट्रॉल? तो you will get cholesterol from the diet, 0.3 gram per day. Cholesterol you will get from the diet. Normally it is present in the butter, milk, cream, butter or milk cream, egg yolk, meat, chicken, egg. Around 60 gram egg will give you 240 milligram cholesterol. मतलब अगर 1 मिलीग्राम का एक अंडा है उसमें 250 मिलीग्राम क्या होता है, कोलेस्टेरॉल होता है। In a 60 milligram egg, chicken egg, you will have 250 gram cholesterol. Cholesterol is also synthesized by body endogenously. It is also synthesized endogenously. It is also synthesized from acetic CoA, only 1 gram per day. अगर बाहर से कोलेस्ट्रॉल आपकी बॉडी में जा रहा है तो सिंथेसिस नहीं होगा। If you are taking cholesterol normally from eggs and meat, okay, so your body will not synthesize. If you are not getting cholesterol from your diet, obviously body will synthesize it. तो आपने कोलेस्ट्रॉल खाया या नहीं खाया कुछ भी फर्क नहीं पड़ता कि अगर आपकी बॉडी कोलेस्ट्रॉल नहीं मिला तो बॉडी का कोलेस्ट्रॉल बनाएगी, आपको मिल रहा है तो बॉडी नहीं बनाएगी। दैट्स इट। कोलेस्ट्रॉल इज important because it has a lot of function in your body, is present in a lot of tissues like brain, cell membrane and all that. Cholesterol is important for our body. आजकल nowadays they are making a lot of noise on this. Occurrence in the humans, humans में कहाँ पे पाया जाता है कोलेस्ट्रॉल? 2% in the brain, 0.3% in the liver, 0.3% in the fatty, 0.2% in the endocrine glands, adrenal glands में 10% or more. So in the human body 2% cholesterol is there in the brain only, 10% is there in the adrenal gland, 0.3% is there in a liver, GIT, 2% in the endocrine glands. So cholesterol is present as either free cholesterol or cholesterol esters. So in a humans cholesterol is present as a free cholesterol or as a esterified with the fatty acid. Now ये esterified जो विद है, esterified with the fatty acid, तो needs a esterification, cholesterol needs a esterification. So esterification of cholesterol in the tissue, esterification occurs in the tissue as well as in the blood, in the plasma as well as in the tissue. So esterification of cholesterol in the tissue, कोलेस्ट्रॉल का esterification मतलब कहाँ पे होता है, टिश्यू में भी होता है और प्लाज्मा में भी होता है। टिश्यू में कहाँ पे होता है, टिश्यू में कौन करता है, तो ट्रांसफर ऑफ असाइल ग्रुप, ट्रांसफर ऑफ असाइल ग्रुप फ्रॉम असाइल CoA to the cholesterol in the tissue. There is esterification of cholesterol in the tissue. Now in the plasma, transfer of acyl group, mostly unsaturated acyl from the lecithin, which is a phospholipid, to the cholesterol by the one enzyme known as LCAT, lecithin cholesterol acyl transferase. So cholesterol acyl transferase enzyme होता है in the tissue that will transfer the cholesterol to the acyl group to the cholesterol. But in the plasma, that is same thing is done by LCAT, lecithin cholesterol acyl transferase. And if there is a genetic deficiency of this enzyme, अगर there is a अगर genetic deficiency होता है lecithin cholesterol If there is a deficiency of this lecithin cholesterol acyl transferase, that leads to Norman's disease. Norman's disease अगर इसका deficiency अगर है Norman's. Now you see the cholesterol structure. यहाँ पे is structure of cholesterol. There are three rings. Rings A. These three rings are known as a phenanthrene ring. Ring D is a cyclopentane ring. So numbering will start from this. Now 1, 2, 3, 4, 5, 1, 8. Then 9, 10. 1, 2, 3, 4, 5, 8, 9 and 11, 12, 13. Right? Right nucleus is a ring structure known as a cyclopentane over hydro. Cyclopentane ring, the phenanthrene ring. So structure of cholesterol, molecular formula C27H. Under the skin, cholesterol is converted into ergosterol. Ergosterol vitamin D is synthesized under the skin. Eicosapolenoic fatty acid. Polenoic fatty acids, जिसमें डबल बॉन्ड ज़्यादा होंगे. Acids from that prostanoids मतलब prostaglandins are made. से आपका arachidonic acid है. That is the eicosapolenoic acid. Prostaglandins, prostacyclins, thromboxane. Also lipoxins and leukotrienes are also made. Again, see the structure of cholesterol, molecular formula of cholesterol C27H45. So this ring system known as a phenanthrene ring system, ring D known as a cyclopentane ring. So cholesterol is having cyclopentane per hydrophenanthrene ring structure. Is a OH at 3 position. Is a double bond between 5 and 6. Okay, is a 2 methyl group at a 2 methyl group at a 18 and 19. At a 17 position, 8 carbon. The other structural feature cholesterol. Now lipoproteins. What is lipoprotein? Lipoprotein क्या होता है? It is a macromolecular complex of lipid protein. अब क्या है? लार्ज साइज का प्रोटीन और लिपिड का कॉम्प्लेक्स. तो प्रोटीन्स होंगे इसमें, दे आर नोन एज अपोप्रोटीन्स. प्रोटीन्स इन द लिपोप्रोटीन नोन एज अपोप्रोटीन. फॉर एग्जांपल अपोप्रोटीन सी, अपोप्रोटीन बी. Apoprotein C is in the periphery, known as a peripheral apoprotein, and this apoprotein B is as a submerged in the structure as it is integrated in the protein structure. Now lipoproteins are made from components like a protein, then phospholipids, then cholesterol. They are arranged in a such a way to look like a sphere. Spherical होता है जैसे लिपोसोम्स ऐसे होते हैं, स्फीयर जैसे look like है ना. And apoprotein C is in a peripheral, integral apoprotein B So the outer part is made from free cholesterol. The core is made from cholesterol esters and triacylglycerol. कोर पार्ट होता है उसमें कोलेस्टेरॉल एंड ट्राइग्लिसराइड. कोर इज मेनली हैविंग नॉन पोलर लिपिड्स, but periphery is having polar lipids. Now we'll see the different lipoproteins composition in a percentage of various lipoprotein. इसके ऊपर क्वेश्चन आता है कौन सा लिपोप्रोटीन में क्या क्या है. So there are different lipoproteins like lipoproteins. Their size is given, triglyceride content in a percentage, cholesterol content in a percentage, phospholipid and protein in a percentage. मतलब लिपोप्रोटीन्स में कंपैरेटिवली कौन से लिपोप्रोटीन्स में ट्राइग्लिसराइड्स कितने हैं, कोलेस्ट्रॉल परसेंटेज में कितना है, फॉस्फोलिपिड कितने हैं, प्रोटीन कितना है. तो फर्स्ट इज अ काइलोमाइक्रॉन्स. Chylomicrons. So it is having highest size 500 mm. Then 500 micrometer. Triglycerides is around 98%. Cholesterol is in a minus, phospholipids in a minus, protein is a 2%. इस काइलोमाइक्रोन में सबसे ज़्यादा क्या होता है, ट्राइग्लिसराइड्स. ट्राइग्लिसराइड्स इज देयर इन अ काइलोमाइक्रोन्स. cholesterol null, phospholipid null, and lipoproteins, proteins 2%. Now very low density lipoprotein, very low density lipoprotein, VLDL, very low density VLDL. So in a size 43 micrometer, triglycerides like null, but cholesterol and phospholipids are present 10% respectively. 10% protein is there. Next one is LDL, low density lipoprotein. They are defined based upon the density. So lipoprotein, low density lipoprotein, 22 micrometer in a size, 15% triglyceride content, 3% cholesterol content. That's why low density lipoprotein, they are known as a bad cholesterol. But they are known as a bad. Phospholipid 25% and protein 20%. High density lipoprotein HDL. High density lipoprotein में 8 micrometer size, 10% triglyceride, 30% cholesterol, 60% phospholipid, 50% protein. So if you compare a low density lipoprotein and high density lipoprotein, तो LDL में 60% कोलेस्ट्रॉल होता है और HDL में ओनली 30%. बट LDL में 60% फॉस्फोलिपिड्स होते हैं जो बेनिफिशियल होते हैं. दैट्स व्हाई LDL इज अ बैड कोलेस्ट्रॉल, HDL इज अ गुड कोलेस्ट्रॉल. HDL क्या है, गुड कोलेस्ट्रॉल है. And again, because of their functions that we are going to see in the next slide, तो इनके जो फंक्शन है जो अगले स्लाइड में देखने वाले हैं, because of their function also you will came to know why HDL is good, why LDL is a bad cholesterol. Now in the chylomicron mostly the triglycerides, the VLDL, again the cholesterol, the intermediate density lipoprotein, there is a again, but in the LDL there is a high level of cholesterol. In the LDL, the level of phospholipid. में हो मैं कॉल करता हूँ आपको 8 बजे. तो मैं लेक्चर में हूँ, मैं 8 सवा 8 बजे Now color reactions of sterol. Color reactions, how to identify this for cholesterol. So very important test and multiple times इसके ऊपर क्वेश्चन आ चुका है. कलर रिएक्शंस ऑफ sterol. पहला टेस्ट है आपका लीबरमन बर्चर्ड टेस्ट. लीबरमन बर्चर्ड रिएक्शन और लीबरमन बर्चर्ड टेस्ट. A green color or green grass color is obtained when this cholesterol or sterol treated in a chloroform. Solution of cholesterol in a chloroform when treated with the acetic anhydride and concentrated sulfuric acid, you will get a green grass color. Green grass color कब आता है? जब कोलेस्ट्रॉल क्लोरोफॉर्म में एसिटिक अनहाइड्रेट एंड कॉन्सनट्रेटेड सल्फ्यूरिक एसिड के साथ में ट्रीट किया जाता है, तो आपको ग्रीन ग्रास कलर आएगा. दैट इज अ कंफर्मेशन फॉर लीबरमन बर्चर टेस्ट. एंड आल्सो इट इज अ basis of estimation of cholesterol by Sackett method. So estimation of cholesterol is done by one of the method known as Sackett method, and ये Lieberman Burchard reaction is the basis of that. तो इसके ऊपर multiple times question आ चुका है. Lieberman Burchard reaction, Lieberman Burchard test. So it is a test to identify the terpenoids also and cholesterol levels. Second test is also there, Salkowski test. Second test is also there, Salkowski. Here red to purple color is obtained when cholesterol or sterols solution in the chloroform treated with the equal volume of sulfuric. तो यहाँ पे भी स्टेरॉइड मतलब कोलेस्ट्रॉल जब क्लोरोफॉर्म में सॉल्यूशन में आप लेंगे, इक्वल वॉल्यूम ऑफ sulfuric acid ऐड करेंगे, you will get a red to purple color. That is the Salkowski test. Now another test is the Zach's reaction. Zach. Cholesterol in the glacial acetic acid, which is free from aldehyde, so it is treated with the ferrichloride and concentrated H2SO4. You will get again a red color. Functions of proteins. पहला है आपका पहला लिपोप्रोटीन कौन सा है? काइलोमाइक्रोन. व्हाट इज द फंक्शन ऑफ दिस लिपोप्रोटीन्स? मोस्टली दे आर द कैरियर्स, दे आर द ट्रांसपोर्ट प्रोटीन. यहाँ पे याद रखना है फंक्शंस ऑफ फंक्शंस ऑफ दिस लिपोप्रोटीन्स, दे आर द कैरियर प्रोटीन. सो काइलोमाइक्रॉन्स दे आर द कैरियर फॉर एक्सोजिनस ट्राइग्लिसराइड. मतलब आप जो ट्राइग्लिसराइड खाते हो या फिर जो फैट्स लेते हो ना, व्हाट एवर लिपिड्स यू आर ईटिंग, तो काइलोमाइक्रॉन्स दे कैरी द एक्सोजिनस ट्राइग्लिसराइड and also in a small amount phospholipids and cholesterol esters and fat soluble vitamins from liver and from intestine to the liver. मतलब chylomicrons are responsible for the absorption of lipids, absorption of triglycerides, phospholipids, cholesterol esters and fat soluble vitamins. So they carries these lipids from intestine to liver. सेकेंड है आपका VLDL, मतलब वेरी लो डेंसिटी लिपोप्रोटीन. VLDL मतलब क्या? वेरी लो डेंसिटी लिपोप्रोटीन. ओके, so again as I told you, these all are the carriers, they are the carriers. जैसे chylomicron is a carrier for triglyceride and esters of cholesterol from intestine to liver. So VLDL is opposite. VLDL, very low density lipoprotein, is a carrier of endogenous, कैरियर फॉर एंडोजेनस. जो बॉडी के अंदर जो है उसको एंडोजेनस. एक्सोजेनस मतलब क्या? बॉडी से बाहर से जो आ रहा है उसको एक्सोजेनस. तो VLDL इज एंडोजेनस है ना, कैरियर फॉर एंडोजेनस ट्राइग्लिसराइड्स है ना. ट्राइग्लिसराइड सिंथेसाइज फ्रॉम इन द लिवर टू द अदर टिश्यू. मतलब लीवर से दूसरे टिश्यूज में, जैसे आपका लीवर से आपका ब्लड वेसल्स, ब्रेन, जो भी टिश्यू है आपका, किडनी और है. तो VLDL will carry triglyceride but from liver to the tissues. Chylomicrons will carry an exogenous triglyceride to the liver. Now, third you can see LDL, low density lipoprotein. Low density lipoprotein. It is a rich in the cholesterol and cholesterol esters. They go rich in the cholesterol and cholesterol esters. And transport cholesterol to extra hepatic tissue. मतलब क्या? ये लीवर से लेके कोलेस्ट्रॉल एंड कोलेस्ट्रॉल एस्टर्स बाकी टिश्यूज में डिपॉजिट कर देगा. जैसे it will take a cholesterol from liver and deposit into the artery, and that can leads to atherosclerosis. That's why it is known as a bad cholesterol, because it deposits cholesterol into the carries cholesterol from liver to the other tissue. That's why bad cholesterol. Now LDL carries the cholesterol from blood vessel and other tissue to the liver. मतलब ये tissues to the liver, the breakdown. मतलब कोलेस्ट्रॉल LDL भी कैरी करता है. LDL का HDL का काम क्या है? टू ट्रांसपोर्ट द कोलेस्ट्रॉल. LDL transport the cholesterol liver to the tissues, which is dangerous, which leads to atherosclerosis. That's why LDL is known as a bad cholesterol. But HDL carries the cholesterol from tissues, peripheral tissues to the liver. That's why it is a good cholesterol. Good cholesterol. Now 6 steps for lipid absorption. Lipid का absorption कैसे होता है? So fats and oils are absorbed. There is a minor digestion of triglycerols in the mouth and stomach. आपका जो माउथ है, सलाइवा है, स्टमक जो है, इसमें जो एंजाइम्स है, माइनर डाइजेशन ऑफ triglycerols by lignal acid stable lipase, the enzyme that breaks lipids. Major digestion that occurs the digestion of all lipids in the lumen of duodenum. तो जो intestine है वहाँ पे major digestion. Duodenum digestion by pancreatic. So again absorption of lipids start from the saliva itself. salivary lipase होता है ना. बाद में pancreatic lipase that absorb the lipids from duodenum or intestine. Then bile acids, they facilitate the formation of micelles, and they get these micelles, they are absorbed or transported through a lipidic system. Bile facilitate the formation of mixed. There is also passive absorption of lipolytic product from mixed micelles to the intestinal. After absorption in the intestinal wall, the re-esterification of monoacyl fatty acids inside the intestinal cell. Then assembly. Chylomicrons containing apoproteins, triglyceride ester and phospholipid, they are and export from the intestinal. This is how there is a absorption of lipids. Absorption कैसे होता है? They are not going to ask you question on this, but should know. Physiologically important lipases. What are the different lipases enzymes? For example, first, sublingual acid stable lipase. It is found in the mouth and stomach. Triglyceride with the medium chain fatty acid, they are peppered. So they are broken down. Converted into free fatty acid and diacylglycerol. So pancreas में जो lipase है, pancreatic lipase and colipase present in the small intestine, act on a site, act at a small intestine, convert triglyceride and long chain fatty acid into free fatty acid and 2 monoacylglycerol. Intestinal lipase with the bile acid. Bile acid के साथ में small intestine में क्या करता है? triglyceride and medium chain fatty acids, they are broken down into 3 fatty acids plus glycerol. So phospholipase A2 is present act on a small intestine. Phospholipase A2 with the unsaturated fatty acid, we have seen the position, convert into unsaturated free fatty acid plus lysoleucine. Lipoprotein lipase in a capillary's wall, triglycerides and chylomicrons or VLDL is broken down into fatty acid. And hormone sensitive lipase जो होता है act on adipose tissue, triglycerides adipose tissue broken down into fatty acid and glycerol. These are the different lipases. lipase जो एंजाइम से ब्रेक डाउन करते, what is the breakdown product? Absorption of lipids. So absorption of fatty acids, long chain fatty acid, देखो long chain are absorbed into the internal wall. They are re-esterified, made into chylomicron and enters into the lymphatics. And short chain fatty acids are directly absorbed into the blood capillaries. Bile acids are reabsorbed total weight. मतलब लॉन्ग चेन फैटी एसिड अगर है, लॉन्ग चेन फैटी एसिड्स है, they are absorbed into the intestinal wall, है ना, absorb होने के बाद में they are esterified with the monocyclic. They are so through lactal tube in the lymphatic system. Small chain or short chain fatty acid, they are absorbed directly into the bile released is by portal vein. So first of all, these bile salts are there: sodium glycocolate, sodium taurocolate. They convert lipid droplet into a emulsion which up into lymphatic system. That's how it is absorbed. The partial hydrolysis, triglyceride, partial hydrolysis, triacyl glycerols, triglycerols, lipase and convert into triacyl. Lipase will act and metabolism of lipids. So metabolism of lipids is most important reaction is oxidation of fatty acid. Beta oxidation of fatty acid is beta oxidation of fatty acid, known as a beta oxidation of fatty acid. तो मेटाबॉलिज्म में ये मेन साइकिल कौन सा है? oxidation of fatty acid. Oxidation of fatty acid on a beta carbon atom which results in the sequential removal of 2 carbon fragments of acetyl CoA. Oxidation of fatty acid on a beta carbon that releases the 2 carbon fragments. Now this beta oxidation of fatty acid is carried out in a 3-step. So what are these 3 steps? First is the activation of fatty acids, cytosol. Cytosol जो है cytoplasm जो है सेल का क्या होता है? Activation of fatty acid. Beta oxidation of fatty acid basically क्या है? It is one of the most important mechanism or metabolism of fatty acid. So oxidation of fatty acid on beta carbon result into the sequential removal of 2 carbon atom fragments CoA. लिपिड कैसा है, कोई। तो ऑक्सीडेशन होने के बाद में क्या मिलेगा आपको, एसिड CoA will enter into Krebs cycle, further utilized, or acetyl CoA can be utilized. We have already seen beta of acetyl CoA, carbohydrate. So activation of fatty acids occurs in the cytosol, then transport of that activated fatty acid by carrier system carnitine. Third, the oxidation of fatty acid. So they will ask you question like that. Beta oxidation of fatty acid occurs in the mitochondria. माइटोकॉन्ड्रिया में होता है, बट एक्टिवेशन साइटोसोल में होता है, ट्रांसपोर्ट होगी माइटोकॉन्ड्रिया में प्रॉपर बीटा ऑक्सीडेशन ऑफ फैटी एसिड सी टुमारो, कल प्रॉपर बीटा ऑक्सीडेशन ऑफ फैटी एसिड्स देखेंगे। देन वहाँ पे उसका बायोसिंथेसिस ऑफ कोलेस्ट्रॉल भी देखेंगे, बिकॉज पीपल आर नॉट ज्वाइनिंग, सो दैट्स व्हाई आई एम ओनली 1 आवर। अगर आप ज्वाइन करते हो तो प्रॉपर 1.5 आवर लेक्चर होगा। दैट्स इट टुमारो। फटाफट ज्वाइन नहीं करते ना।